Road to Taiwan presents the first year medical topic review. Cell. Did you know that cell is the basic unit of life? This small cell is actually the one who works day and night to help you stay awake in the morning and asleep in the evening. It is the soldier of your body when you need to fight infections. It signals other cells to provide function needed by your body. Your body fluids is also composed of cells, for example your blood. This one cell you see right here has small compartments, each with different functions. The first and foremost important that you should remember is the nucleus, which is center of control in the cell where the genome of your body is located. There is a mitochondria which functions for ATP synthesis by oxidative phosphorylation. Endoplasmic reticulum, there are two, smooth and rough, both is used for protein translation. Meanwhile this free ribosomes found in the rough endoplasmic reticulum is for protein synthesis. There are some apparatuses called Golgi bodies used for modification, packaging and sortage of proteins to be secreted in and out of the cell. Lysosomes, peroxisomes and proteasomes are used for intracellular degradation of foreign bodies that may affect the cell. Second major part of the cell is the plasma membrane, which covers the cell. Third is the cytosol, where organelles talked about earlier are located. Let us focus at the nucleus. You can think of the cell nucleus as a kind of command center, as I said earlier, containing all the chromosomal and genetic information needed for the reproduction of life. It is bound by a double membrane called the nuclear envelope. This membrane separates the contents of the nucleus from the cytoplasm. Like the plasma membrane, the nuclear envelope consists of phospholipids that form a lipid bilayer. The envelope helps to maintain the shape of the nucleus and assists in regulating the flow of molecules into and out of the nucleus through nuclear pores. The nuclear envelope is connected with the endoplasmic reticulum, ER, in such a way that the internal compartment of the nuclear envelope is continuous with the lumen of the ER. The nucleus is the organelle which houses chromosomes. These chromosomes consist of DNA, which contains heredity information and instructions for cell growth, development, and reproduction. When a cell is resting, or not dividing, the chromosomes are organized into long entangled structures called chromatin and not into individual chromosomes as we naturally think of them. Nucleoplasm is the gelatinous substance within the nuclear envelope. Also called karyoplasm, this semi-aqueous material is similar to cytoplasm and is composed mainly of water with dissolved salts, enzymes, and organic molecules suspended within. Contained within the nucleus is a dense membrane, hyphenless structure composed of RNA and proteins called the nucleolus that contains nuclear organizers, which are parts of chromosomes with the genes for ribosome synthesis on them. Next is the Golgi apparatus. The Golgi apparatus, or complex or body HISTO, plays an important role in the modification and transport of proteins within the cell. It is called the manufacturing and shipping center of the eukaryotic cell. The Golgi apparatus, sometimes called the Golgi complex or Golgi body, is responsible for manufacturing, warehousing, and shipping certain cellular products, particularly those from the endoplasmic reticulum, ER. Depending on the type of cell, there can be just a few complexes or there can be hundreds. Cells that specialize in secreting various substances typically have a high number of Golgi. That contains cisternae. Cisternae are flat sacs that are stacked in a semicircular, bent formation. Each formation has a membrane to separate it from the cytoplasm of the cell. The Golgi apparatus has several functions, including modification of several products from the endoplasmic reticulum, ER. Examples include phospholipids and proteins. The apparatus can also manufacture its own biological polymers. The Golgi complex is capable of both disassembly and reassembly during mitosis. In the early stages of mitosis, it disassembles while it reassembles in the telophase stage. Powered by converting energy into forms that are usable by the cell. Are the sites of cellular respiration, which is a process that ultimately generates fuel for the cell's activities from the foods we eat. Mitochondria produce the energy required to perform processes such as cell division, growth, and cell death. This has a distinctive oblong or oval shape and are bounded by a double membrane. The inner membrane is folded, creating structures known as cristae. They are found in all body cell types, except for mature red blood cells. As mentioned, red blood cells do not contain mitochondria at all. The absence of mitochondria and other organelles in red blood cells leaves room for the millions of hemoglobin molecules needed in order to transport oxygen throughout the body. Muscle cells, on the other hand, may contain thousands of mitochondria needed to provide the energy required for muscle activity. They are also abundant in fat cells and liver cells. Within the inner mitochondrial membrane are a series of protein complexes and electron carrier molecules, which form the electron transport chain, ETC. The ETC represents the third stage of aerobic cellular respiration and the stage where the vast majority of ATP molecules are generated. ATP is the body's main source of energy and is used by cells to perform important functions, such as muscle contraction and cell division. Look at this. This is called endoplasmic reticulum. It works closely with the Golgi Apparatus, ribosomes, RNA, and RNA. Structurally, this is a network of membranes found throughout the cell and connected to the nucleus. The membranes are slightly different from cell to cell, and a cell's function determines the size and structure of the ER. There are two basic types of ER. These are rough ER and smooth ER. Both have the same types of membranes, but they have different shapes. Rough ER looks like sheets or disks of bumpy membranes, while smooth ER looks more like tubes. Rough ER is called rough because it has ribosomes attached to its surface. Its double membranes of smooth and rough ER form sacs called cisternae. Protein molecules are synthesized and collected in the cisternal space or lumen. When enough proteins have been synthesized, they collect and are pinched off in vesicles. The vesicles often move to the Golgi apparatus for additional protein packaging and distribution. There are also cytoskeleton, which is composed of at least 3 different types of fibers: microtubules, microfilaments, and intermediate filaments. These fibers are distinguished by their size, with microtubules being the thickest and microfilaments being the thinnest. Let us define each. Microtubules are hollow rods functioning primarily to help support and shape the cell and as roots, along which organelles can move. They are typically found in all eukaryotic cells. They vary in length and measure about 25 nanometers in diameter. Microfilaments or actin filaments are thin, solid rods that are active in muscle contraction. They are particularly prevalent in muscle cells. Similar to microtubules, they are typically found in all eukaryotic cells. They are composed primarily of the contractile protein actin and measure up to 8 in diameter. They also participate in organelle movement. Intermediate filaments can be abundant in many cells and provide support for microfilaments and microtubules by holding them in place. These filaments form keratins found in epithelial cells and neurofilaments in neurons. They measure 10 in diameter. As cytoskeletal microfilaments contract, they help to direct the flow of cytoplasmic particles. When microfilaments attached to organelles contract, the organelles are pulled along and the cytoplasm flows in the same direction. A number of motor proteins are found in the cytoskeleton. But first, what is protein? It is any of a class of nitrogenous organic compounds that consist of large molecules composed of one or more long chains of amino acids and are an essential part of all living organisms, especially as structural components of body tissues such as muscle, hair, collagen, etc., and as enzymes and antibodies. The simplest level of protein structure, primary structure, is simply the sequence of amino acids in a polypeptide chain. The sequence of a protein is determined by the DNA of the gene that encodes the protein, or that encodes a portion of the protein, for multisubunit proteins. A change in the gene's DNA sequence may lead to a change in the amino acid sequence of the protein. Even changing just one amino acid in a protein sequence can affect the protein's overall structure and function. The next level of protein structure, a change in the gene's DNA sequence may lead to a change in the amino acid sequence of the protein. Even changing just one amino acid in a protein sequence can affect the protein's overall structure and function. The next level of protein structure, secondary structure, refers to local folded structures that form within a polypeptide due to interactions between atoms of the backbone, which refers to the polypeptide chain apart from the R groups. So all we mean here is that secondary structure does not involve R group atoms. The most common types of secondary structures are the helix and the pleated sheet. These structures are held in shape by hydrogen bonds, which form between the carbonyl O of one amino acid and the amino H of another. The overall three-dimensional structure of a polypeptide is called its tertiary structure, which is primarily due to interactions between the R groups of the amino acids that make up the protein. These interactions that contribute to tertiary structure include hydrogen bonding, ionic bonding, dipole-dipole interactions, and London dispersion forces. Some proteins are made up of multiple polypeptide chains, also known as subunits. When these subunits come together, they give the protein its quaternary structure. In general, the same types of interactions that contribute to tertiary structure, mostly weak interactions, such as hydrogen bonding and London dispersion forces, also hold the subunits together to give quaternary structure. Each protein has its own unique shape. If the temperature of the protein's environment is changed, or if it is exposed to chemicals, these interactions may be disrupted, causing the protein to lose its three-dimensional structure and turn back into an unstructured string of amino acids. When a protein loses its higher-order structure, but not its primary sequence, it is said to be denatured. Denatured proteins are usually non-functional. Let us discuss the central dogma of molecular biology, which describes the two-step process, transcription and translation, by which the information in genes flows into proteins. DNA RNA protein starts with replication, then transcription, which is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme RNA polymerase. Translation begins while the RNA is still being synthesized. In a eukaryotic cell, transcription occurs in the nucleus, and translation occurs in the cytoplasm. In prokaryotic cell, because there is no nucleus to separate the processes of transcription and translation, when bacterial genes are transcribed, their transcripts can immediately be translated. In eukaryotic cell, transcription and translation are spatially and temporally separated in eukaryotic cells, that is, transcription occurs in the nucleus to produce a pre-RNA molecules. The pre-RNA is typically processed to produce the mature RNA, which exits the nucleus and is translated in the cytoplasm. DNA is made up of 6 smaller molecules, a 5-carbon sugar called deoxyribose, a phosphate molecule, and 4 different nitrogenous bases, adenine, thymine, cytosine and guanine. There are hydrogen bonds between complementary bases. These are the structures of B and Z DNA. When a cell divides, each daughter cell must receive its full complement of genetic material in the form of chromosomes containing DNA, or deoxyribonucleic acid. A chromosome is made up of 2 long strands of DNA and several types of proteins. A DNA strand is composed of a long backbone of sugar and phosphate units. One of our different nucleotide bases, -A, T, C or G, hang off each sugar unit. The sequence of the bases encodes genetic information. The 3 steps in the process of DNA replication are initiation, elongation and termination. Replication depends on the pairing of bases between the 2 strands of DNA. The A base can only bind to a T, and a C can only bind to a G in the DNA double helix. The bases of one strand face across and bind to those of the other strand. Therefore, the base sequence of each strand complements that of the other. The sequences are antiparallel and serve as templates for each other's replication. The 2 strands are labeled by the location of certain chemical bonds in the DNA backbone. The cell can replicate the leading strand, as a single unit, but must replicate the lagging strand, in small pieces. Replication begins at a location on the double helix known as ORIC, to which certain initiator proteins bind and trigger unwinding. Enzymes known as helicases unwind the double helix by breaking the hydrogen bonds between complementary base pairs, while other proteins keep the single strands from rejoining. The topoisomerase proteins surround the unzipping strands and relax the twisting that might damage the unwinding DNA. The cell prepares for the next step, elongation, by creating short sequences of RNA called primers that provide a starting point of elongation. With the primer as the starting point for the leading strand, a new DNA strand grows one base at a time. The existing strand is a template for the new strand. For example, if the next base on the existing strand is an A, the new strand receives a T. The enzyme DNA polymerase controls elongation, which can occur only in the leading direction. The lagging strand unwinds in small sections that DNA polymerase replicates in the leading direction. The resulting small Okazaki fragments can contain 1,000 to 2,000 bases in bacteria, but eukaryotes, organisms having cells with nuclei, have fragments of only 100 to 200 bases. The fragments terminate in an RNA primer that is subsequently removed so that enzymes can stitch the fragments into an elongating strand. After elongation is complete, two new double helices have replaced the original helix. During termination, the last primer sequence must be removed from the end of the lagging strand. This last portion of the lagging strand is the telomere section, containing a repeating non-coding sequence of bases. Enzymes snip off a telomere at the end of each replication, leading to shorter strands after each cycle. Finally, enzymes called nucleases proofread the new double helix structures and remove mispaired bases. DNA polymerase then fills in the gaps created by the excised bases. Oxidative phosphorylation is the metabolic pathway in which cells use enzymes to oxidize nutrients, thereby releasing energy which is used to produce adenosine triphosphate. You, like many other organisms, need oxygen to live. As you know if you've ever tried to hold your breath for too long, lack of oxygen can make you feel dizzy or even black out, and prolonged lack of oxygen can even cause death. But have you ever wondered why that's the case, or what exactly your body does with all that oxygen? As it turns out, the reason you need oxygen is so your cells can use this molecule during oxidative phosphorylation, the final stage of cellular respiration. Oxidative phosphorylation is made up of two closely connected components, the electron transport chain and chemiosmosis. In the electron transport chain, electrons are passed from one molecule to another, and energy released in these electron transfers is used to form an electrochemical gradient. In chemiosmosis, the energy stored in the gradient is used to make ATP. Oxygen sits at the end of the electron transport chain, where it accepts electrons and picks up protons to form water. If oxygen isn't there to accept electrons, for instance, because a person is not breathing in enough oxygen, the electron transport chain will stop running, and ATP will no longer be produced by chemiosmosis. Cells without ATP can't function. The electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. Electrons are passed from one member of the transport chain to another in a series of redox reactions. First there is a delivery of electrons by ANAT and FAT, wherein reduced electron carriers from other steps of cellular respiration transfer their electrons to molecules near the beginning of the transport chain. In the process, they turn back into ANAT and FAT, which can be reused in other steps of cellular respiration. Electron transfer and proton pumping is done. As electrons are passed down the chain, they move from a higher to a lower energy level, releasing energy. Some of the energy is used to pump H++ ions, moving them out of the matrix and into the intermembrane space. This pumping establishes an electrochemical gradient. Splitting of oxygen to form water is happening. At the end of the electron transport chain, electrons are transferred to molecular oxygen, which splits in half and takes up H++ to form water. There is a gradient-driven synthesis of ATP. As H+ ions flow down their gradient and back into the matrix, they pass through an enzyme called ATP synthase, which harnesses the flow of protons to synthesize ATP. All of the electrons that enter the transport chain come from NAD and FAT molecules produced during earlier stages of cellular respiration, glycolysis, pyruvate oxidation, and the citric acid cycle. Beyond the first two complexes, electrons from NAD and FAT travel exactly the same route. Both complex I and complex II pass their electrons to a small, mobile electron carrier called ubiquinone, Q, which is reduced to form Q and travels through the membrane, delivering the electrons to complex third. As electrons move through complex 3, more H+ ions are pumped across the membrane, and the electrons are ultimately delivered to another mobile carrier called cytochrome C, CytC. CytC carries the electrons to complex IV, where a final batch of H+ ions is pumped across the membrane. Complex IV passes the electrons to O, which splits into 2 oxygen atoms and accepts protons from the matrix to form water. 4 electrons are required to reduce each molecule of O, and 2 water molecules are formed in the process. What does the electron transport chain do for the cell? It has 2 important functions. Regenerates electron carriers and makes a proton gradient. Fluid can enter the body as performed water, ingested food and drink, and, to a lesser extent, as metabolic water that is produced as a byproduct of aerobic respiration and dehydration synthesis. A constant supply is needed to replenish the fluids lost through normal physiological activities, such as respiration, sweating, and urination. Water generated from the biochemical metabolism of nutrients provides a significant proportion of the daily water requirements for some arthropods and desert animals, but it provides only a small fraction of the humans.